Roger Albin. I'm a neurologist at the Alzheimer's Center at the University of Michigan. And we're going to talk today about molecular imaging of neurodegenerative dementias. Now, molecular imaging has become the preferred term in comparison to the older term, which was positron emission tomography. And molecular imaging is appropriate because the goal here is actually to quantify the distribution and density of specific molecules within an organ of interest, in this case, the brain. And these methods have become extremely important in clinical research regarding dementing disorders and have begun to intrude into clinical practice as well. These are my disclosures, none of which are relevant to today's presentation. This is an outline of what we're going to discuss. Our overall goal is to understand at a basic level the role of PET imaging in dementia research. And then we're going to cover PET imaging of Alzheimer's disease, Lewy body dementias, frontotemporal dementias, and also proteinopathy imaging. It's important to specify some important regulatory information. Some of the tracers discussed today are approved by the FDA for clinical use. These include fluorodeoxyglucose, FPCIT, and florbetapir. FPCIT is known commercially as DAT-scan, is actually a SPECT imaging method, which is a related method to PET imaging, and florbetapir is known commercially as Amyloid-Avid. All the other radio tracers I'm going to discuss are used for investigational purposes only and are not approved by the FDA for clinical use. Fluorodeoxyglucose is reimbursed by the Centers for Medicare and Medicaid Services, but only for the distinction of frontotemporal dementias versus Alzheimer's disease. And FPCIT is approved for the distinction of Parkinsonism versus essential tremor. Florbetapir is approved for detection of patholoid amyloid deposition in the brain. Now, much of the audience will be familiar with this, but this is just to briefly outline the importance of dementia epidemiology and its social impact. So a reasonable prevalence estimate would be about 1,400 per 100,000 in the United States. And this would translate to about 4.5 million people who are affected in the United States. And as this audience knows, this is an age-related condition. So one reasonable estimate is that perhaps 4 to 5% of individuals in the 70 to 80 decade experience dementing illness, and that rises to as many as a quarter of subjects in the age range of 80 to 90. This is an extremely costly illness. A reasonable estimate derived several years ago by my colleague here, Kenneth Langa, estimated that actual annual costs of dementing illnesses was in the order of $150 billion. And this is comparable to the costs of cancer care and cardiovascular disease in the United States. Now, it's important to specify also that dementia is really a meta-syndrome. It's an umbrella term covering a number of different dementing syndromes. The most common of these is thought to be Alzheimer's disease, and followed by perhaps vascular dementia around 20%, dementia with Lewy bodies perhaps another 20%, and frontotemporal dementias in the order of 5% or less. Important stipulation, however, is that in actual fact, mixed dementia is really the most common pathology found in elderly demented individuals. There's usually a spectrum of different age-related pathologies, and it's often difficult to determine what's the dominant pathology contributing to any given individual's dementing clinical features. A major feature, though, is that our routine clinical diagnostic accuracy, at least as compared with pathologic examination, is relatively poor. So in a variety of studies, diagnostic clinical accuracy is on the order of 60 to 80%. In addition to just the problems of accurate diagnosis in symptomatic patients, we really have a need to identify early to pre-symptomatic disease, because this is the phase of the illness that we'd really like to be able to target for interventions to slow or even abate the progression of dementing illness. Now, it's important to stipulate a few basic features of neurodegenerations. One basic point is that these are syndromes, not specific diseases. They generally don't have a single specific etiology. They're usually either idiopathic or most likely multifactorial in origin. Exceptions would be some genetic disorders such as Huntington's disease, spinocerebellar ataxias, or some Mendelian forms of Alzheimer's disease. Another basic feature is that we're usually defining our dementias on the basis of neuropathology, but this usually represents some form of disease end state and may not tell us very much about the initial pathophysiologic events that triggered or drive the neurodegenerative processes. And it's those early events that we really are interested in understanding, because those are the avenues for effective intervention. Now, the presence of abnormal protein deposits is used as the primary pathologic criterion for defining these different neurodegenerative disorders. And in terms of imaging targets, we're really talking about three major categories of protein deposits. One of which would be alpha-synuclein, which is seen in Parkinson's disease and multiple system atrophy. A second one would be A-beta peptide deposition, seen in Alzheimer's disease and also in amyloid angiopathy. A third would be tau protein, where the protein deposits are found in Alzheimer's disease, but also in frontotemporal dementias, as well as rare neurodegenerative syndromes such as progressive supranuclear palsy and corticobasal degeneration. And of course, you can have overlapping and co-occurring pathology. So a fair number of individuals with dementia with Lewy bodies also exhibit a significant amount of Alzheimer's pathology as well. This slide provides a capsule summary of the basic underlying principles of positron emission tomography, or PET. Now on your left is a famous painting by the Belgian surrealist painter René Magritte. And I included this image to emphasize that positron emission tomography is based on emission. Now, unlike regular X-ray methods where the radiation source is outside the organ of interest, in PET scanning, the radiation source is inside the organ of interest. Hence, it's an emission method. And the right image gives you a brief capsule of how this works. Now, the starting point is that you have a probe that can bind with high specificity and high affinity for some target in the brain. And that target can be a specific protein, it can be an enzyme, it can be a neurotransmitter receptor. But you have some kind of molecule that will get through the blood-brain barrier and bind with high affinity, and we hope some specificity, to the molecule that you're interested in imaging. And then that molecule is then tagged, that tracer is then tagged with a positron-emitting radioisotope. And the positron-emitting radioisotope undergoes beta decay and emits a positron. And then it interacts with a nearby electron and undergoes a mutual annihilation event, and that generates two orthogonal photons. These are, their path is 180 degrees out of phase. And then your organ of interest is surrounded by a ring of detectors. And essentially, when photons pass through the tissue and through the skull, they hit the detectors within a defined time frame. They can be detected as significant coincident events. And based on using some fancy math, you can reconstruct from these coincident events the distribution of the binding site and its density in the tissue of interest, in this case in the brain. So very frequently, the limiting factor is making a probe that you can label with a positron-emitting radioisotope, a tracer that will bind with the appropriate specificity and affinity to the target that you're interested in. We're now going to discuss what has been the three major innovations in using PET as applied to dementia research. And the first of these has been imaging synaptic density and function using F18 fluorodeoxyglucose PET, and more recently another allied method called SV2A PET. A second line of research has been imaging specific pathologic features of a specific syndrome, and that specifically refers to dementia with Lewy bodies, and we'll talk about the use of dihydrotetrabenazine DTBZ PET and related methods in this context. And finally, a lot of important work that's been done in trying to image characteristic proteinopathies, both A-beta amyloid and tau protein. We're now going to discuss the deoxyglucose method that is the basis for fluorodeoxyglucose PET imaging. And this method is fundamentally a measure of regional brain activity. Now, the brain essentially runs on glucose. Under some conditions, notably starvation, it can run on alternative energy substrates, but under normal conditions, the brain is highly dependent on glucose metabolism for its energy. And the key feature here is that glucose enters brain tissue and undergoes phosphorylation to glucose 6-phosphate, and then it proceeds down the glycolysis pathway and ultimately into the Krebs cycle and for metabolism. But if you administer an analog of glucose, deoxyglucose, what happens is that the deoxyglucose undergoes the initial step, which is phosphorylation, but then it gets stuck. It cannot be metabolized farther down the pathway. And you can label deoxyglucose either with an isotope such as C14, which is used for animal studies, or you can use F18, the positron emitting isotope, which is used for human work. And then the uptake of the fluorodeoxyglucose will be a measure of brain regional glucose uptake and a measure of brain glucose metabolism. Now, a key point is that brain energy metabolism under resting conditions is largely driven by the need to restore ionic gradients after action potential generations. This is an energetically extremely costly activity. And under normal circumstances, regional glucose uptake is a function of how much of synaptic density there is in that volume of tissue. So you can use FDG uptake, fluorodeoxyglucose uptake, as a proxy for synaptic density or synaptic integrity. This is a set of images of fluorodeoxyglucose uptake in a normal individual. And what you can see is that the fluorodeoxyglucose uptake largely identifies gray matter structures. Again, this is consistent with the fact that you're largely measuring synaptic density, as gray matter has a high density of synapses, whereas white matter has a low density. And you can see on the top 4 rows of this image are transaxial sections showing there are some regional differences in different cortical regions, but overall there's a marked gray matter, white matter differentiation. And the bottom row shows an alternative way of representing this data, which is essentially a surface rendering of the activity of glucose uptake. Application of fluorodeoxyglucose PET revealed characteristic abnormalities in Alzheimer's disease. And what's particularly seen is reduced metabolism, reduced fluorodeoxyglucose uptake in the posterior cingulate and precuneus cortex, deficits of uptake in the temporoparietal association cortex and prefrontal association cortices. And in the same individuals, when there's preserved metabolism in primary somatomotor cortex, primary visual cortex, and subcortical structures such as the basal ganglia thalamus, and also structures within the posterior fossa. Now, these metabolic deficits may be asymmetrical, that is, to differ somewhat from hemisphere to hemisphere, but they are generally internally consistent. This image depicts typical metabolic deficits in an individual with Alzheimer's disease. And what's seen particularly in these images are temporoparietal hypometabolism, as well as hypometabolism in the posterior cingulate and precuneus region. And you can see these deficits with the overlay showing, which is a contrast between this individual and a normal control database. And in the right column, you can also see the surface renderings, which show very nicely the temporoparietal deficits as well as the posterior cingulate and precuneus deficits. These metabolic deficits are seen not only in individuals with Alzheimer's disease, but they can also be detected prior to the onset of dementia. So this particular image shows metabolic deficits in individuals with mild cognitive impairment. And these type of abnormalities have been detected in individuals some years prior to the probable conversion to a demented state, including individuals who have familial forms of Alzheimer's disease and carriers of the ApoE4 allele. I've been presenting FDG PET really as a measure of synaptic mass or synaptic density, but it's important to put another stipulation into this, in that in theory, You could see the same abnormalities if a synapse was preserved but not functioning at a normal level of activity. And this particular interpretation is supported by some studies with a new, more complementary PET method called SV2A PET. Now, SV2A is a synaptic vesicle protein, which is involved in synaptic vesicle recycling and exocytosis. And in the image you see on the left there, this shows the participation of SV2A in synaptic vesicle function. Now, in a very clever and nice illustration of the way you can use biology and creative chemistry to develop PET tracers, it was discovered that SV2A is the binding site of a commonly used anticonvulsant, levetiracetam, which is the LEV structure you see designated in the image. And a number of derivatives of levetiracetam were made, leading to a compound called UCBJ. And UCBJ was then underwent labeling with C11, a positron-emitting radioisotope. And this was developed as a successful compound to bind to SV2A. Now, SV2A is expressed in virtually all synaptic terminals. So the SV2A binding pattern, I should say the UCBJ binding pattern, is essentially a measure of synaptic terminal density. And you can see in the image here of the PET image of a human brain here, a very nice depiction of gray matter throughout the central nervous system, consistent with this being a marker of synaptic terminals in total. Now, this is a nice study that was done by a group at Yale comparing SV2A PET and FDG PET in the same individuals. And what you can see in the top row is an individual with Alzheimer's disease who was imaged with the SV2A tracer. And the arrow points to an area of decreased tracer binding in the medial temporal lobe, which is consistent with Alzheimer's pathology. Now, in the middle row, you see the results of FDG PET imaging. And again, what you can see is some deficits in tracer uptake in the medial temporal lobe. But in the case of the fluorodeoxyglucose PET, you can see actually the deficit seems to be larger in extent. And what's seen with the FDG, but not with the SV2A tracer, is some deficits in the parietotemporal cortex as well. And what this suggests, in fact, is that the FDG PET results suggest that there's dysfunctional synapses, but the SV2A, being relatively normal in some regions, suggests that those synapses are dysfunctional, but still relatively intact. A more specific imaging measure of Alzheimer's disease, there was considerable effort devoted to trying to find tracers that would identify one of the characteristic pathologic features, which was deposition of A-beta peptide in amyloid plaques. And this cartoon just shows the different routes of metabolism of the amyloid precursor protein, one of which leads to one pathway which generates soluble peptide fragments, but the other leads to the generation of the A-beta peptide, which is the predominant constituent of the characteristic A-beta plaques. Different possible chemical structures were explored as potential tracers to identify brain amyloid deposition. And most of these were derivatives of thioflavin T, a dye that's used by pathologists really for decades to identify amyloids in tissues. And a number of these compounds were pursued as PET tracers, with the initial success coming with the PIB compound or Pittsburgh B compound. It shows some of the initial successful work done with Pittsburgh B, which was developed by Chet Mathis and Bill Klunk at the University of Pittsburgh, hence the designation Pittsburgh compound. Well, what you can see in the image is a control individual where you see quite a bit of tracer uptake, but it's largely restricted to white matter, which is characteristic of this tracer. Then you have an individual with mild cognitive impairment who has some degree of tracer uptake in the cortex, and then an individual who has Alzheimer's disease who has a high density of cortical uptake of the tracer. Exploration of this tracer disclosed some very interesting results. So it was certainly strongly expressed by individuals who were thought on clinical grounds to have Alzheimer's disease. But an unexpected and quite interesting finding was established by groups both in the United States and Australia, which was that significant amyloid deposition was found in a number of individuals who are cognitively normal. So this slide shows one data from one of these studies. What you can see in the top row are individuals with clinical diagnosis of Alzheimer's disease and clearly have a high density of tracer uptake and presumably amyloid deposition in their cortex. And in the middle row, these are cognitively normal elderly individuals who have little or no amyloid burden. But in the bottom row is an example of an individual who is cognitively normal, but this older individual has significant amyloid burden in their cortex. And this may be true of as many as 30% of people over the age of 65. Now, this also is consistent with pathologic data, which demonstrates that there is a fair number of older people who are cognitively intact who at postmortem have a significant amyloid burden. And it's also consistent with the idea that while amyloid might be a triggering factor for the development of Alzheimer's disease, it's not the factor that really drives the neurodegeneration or cognitive impairment. Of course, it was important to establish that this tracer was actually measuring amyloid burden. And this was done in several studies, which weren't easy to do because they required actually studying people who were fairly close to death and in stage of advanced Alzheimer's disease. But these studies were successfully performed, and this is a nice example. So what you can see in the top row, this is a PET image of an individual who had a low amyloid burden. And on the right, this is a histology from that individual showing that individual has very little amyloid detected by pathologic examination. In the middle row, this is an individual who had what was felt to be a moderate amount of amyloid by PET imaging, and indeed, on histopathologic examination, had a moderate amount of amyloid burden. And finally, in the bottom slide, you have an individual with a very high amyloid burden by PET, and indeed a high amyloid burden by histology. And in general, in this study and some other validation studies, there was a good relationship between PET results and postmortem results, providing a lot of assurance that this measure is actually a good measure of amyloid burden. Another use of this kind of PET method is for target engagement study. Now, target engagement is particularly important in developing new therapies because if you're trying a therapy and it fails, you may fail because your therapy isn't actually affecting the pathophysiologic process that you think it is. And without evidence of target engagement, negative results in trials can be quite confusing. So this is a nice example of target engagement for an anti-amyloid therapy. And this is assessing amyloid burden in the brain during therapy with a monoclonal antibody against amyloid called Aducanumab. Now, many of you will recognize this particular antibody as being quite controversial. It was proposed for FDA approval as a therapy and granted a provisional approval, but with quite a bit of controversy because the clinical results were rather confusing. Regardless of the clinical results, however, PET imaging made it possible to demonstrate that there was in fact target engagement with this therapy. And what you can see here in the images on the right is evidence that use of the antibody actually did result in decreased amyloid burden in the brain. And on the left in the graphs, you can show that this was clearly a dose-related effect. So this definitely demonstrated target engagement, so that any negative results obtained with this antibody weren't because it failed to reduce amyloid burden, but rather possibly because the reduction of amyloid burden wasn't actually that germane to the relevant clinical pathophysiology. Let's turn to dementia with Lewy bodies, which may be the second most common of degenerative dementia. There's diagnostic clinical criteria for Lewy body dementia. This is from an older version of the criteria, but the results here are relatively similar in that these criteria stress visual hallucinations, fluctuating mental status, Parkinsonism as major features. And in terms of diagnostic sensitivity, these criteria are not all that wonderful. Their specificity is actually pretty good, but in terms of sensitivity, varying results were obtained. And while the citations here are some older studies, the more recent criteria really don't perform much better. So this gave rise to interest in trying to produce better methods for diagnosis and classification of dementia with Lewy bodies. Not surprisingly, the first method applied was for deoxyglucose PET imaging. And what you can see here is a representative case of an individual with dementia with Lewy bodies and their FDG PET results. And what you'll see is that the FDG PET results, as shown best by the overlay, really resemble in many respects the FDG PET results of Alzheimer's disease. So you see parietotemporal hypometabolism, you can see precuneus hypometabolism, but there is one significant difference. In Alzheimer's disease, there's usually sparing of occipital cortex, whereas in dementia with Lewy bodies, occipital cortex is frequently involved, and the posterior cingulate is also frequently spared. And this is a fairly characteristic pattern which has a very, very good specificity. Now you can probably see this best in the surface renderings in the right column. Unfortunately, while this is a very specific result, it's not very sensitive, and the sensitivity of FDG PET for diagnosis of dementia with Lewy bodies is about the same as the sensitivity for clinical diagnosis. The limitations of both clinical diagnosis and the FDG PET imaging led to an interest in trying to find an alternative imaging method for diagnosis. And since the great majority of patients with dementia with Lewy bodies manifest Parkinsonism, there was interest in using imaging of dopaminergic terminals to see if that would work as a diagnostic tool. And there are several different PET methods which are depicted on this cartoon, which identified dopaminergic nigrostriatal terminals, which would be candidates for such a method. The idea being that if in a dementing illness that would resemble dementia with Lewy bodies or Alzheimer's disease, the presence of dopaminergic terminal deficits would signal that this was dementia with Lewy bodies. And this was a possibility was explored with a number of these different PET methods. This image shows the results of one study looking at dopamine terminal imaging as a method to detect or classify dementia with Lewy bodies. And this uses VMAT2 PET, which is a very good marker of presynaptic nigrostriatal dopamine terminals. And what you can see is NC is the normal control, where you can see a normal distribution of nigrostriatal terminals. Then an individual with Alzheimer's disease, who again has normal nigrostriatal terminal integrity. But then in individuals with dementia with Lewy bodies and Parkinson's disease, you can see there are marked deficits in this particular marker. And both with this method and closely related methods, it's clear that this was a reliable marker of dementia with Lewy bodies. An allied method to this, which uses SPECT imaging, is actually used to some extent in the United States and considerably more in some European countries as a diagnostic tool for establishing a diagnosis of dementia with Lewy bodies. It's possible also that this method may help to differentiate dementia with Lewy bodies from Parkinson's disease, since the dementia with Lewy bodies subjects seem to have more symmetric loss of dopamine terminals. Survey of major forms of neurodegenerative dementia, we'll briefly go over some of the imaging features of frontotemporal dementias. Now, the major problem here is differentiating the behavioral variant of frontotemporal dementia from Alzheimer's disease. And the language dominant forms of frontotemporal dementia, such as primary progressive aphasia, semantic dementia, these seem to be less difficult to differentiate. But even using more recent criteria for dementing disorders, there's a substantial overlap in the outcome of using clinical diagnostic criteria for frontotemporal dementias and Alzheimer's disease. And this, of course, creates significant diagnostic problems. Now, this set of images shows the characteristic FDG abnormalities found with studies of frontotemporal dementias. Now, not surprisingly, given that the pathology in these regions, in these individuals, is found in the frontal cortex and in anterior temporal cortices, this is where we see metabolic deficits. And you can see this best here in the transaxial sections with the overlay. And most prominently in the surface projections in the right column, where you can see clear abnormalities of frontal and anterior temporal metabolism. This slide summarizes the patterns of FDG PET abnormalities in the various degenerative dementia syndromes. So in the top row, you can see the characteristic features in Alzheimer's disease, parieto-temporal hypometabolism, posterior cingulate and precuneus hypometabolism, and frontal cortical hypometabolism. In the second row is the characteristic features of dementia with Lewy bodies. Again, this resembles the features that you see in Alzheimer's disease with the addition of occipital hypometabolism. And in the bottom two rows, you can see the abnormalities that are characteristic seen in frontotemporal dementias. In some individuals, this is dominated by prefrontal hypometabolism and others by anterior temporal hypometabolism. In general, these abnormalities are quite characteristic and are actually useful in clinical practice for evaluating dementia in uncertain situations. Extension of methods to classify dementias with PET methods would be to try and use some kind of multiple method or multi-tracer classification system. And this has been explored by a number of different groups, and this is just an example from a study that we did in individuals with early dementia or mild cognitive impairment. And what we suggested trying was to try to classify individuals on the basis of both their amyloid deposition and also their nigrostriatal terminal integrity. And this image shows the way classification were done on the basis of imaging studies. So you could see an individual is classified as Alzheimer's disease, had high amyloid burden, but normal nigrostriatal dopamine terminal density. An individual with dementia with Lewy bodies could have normal amyloid burden or somewhat increased amyloid burden, but had to have a marked decrease in their nigrostriatal terminal density. And we classified individuals as having frontotemporal dementia if they did not have amyloid burden, but could have either normal nigrostriatal terminal density or some mild reduction. This last point constitutes a little bit of a weakness of this approach, as there are some individuals with frontotemporal dementia who will develop Parkinsonism and have significant nigrostriatal terminal deficits. This slide shows the results of this study involving almost 100 subjects. And what you see, this is a matrix where we assume that the imaging classification is the proverbial gold standard and compare it against the clinical classification. And what you can see in the right-hand column is the kappa scores. This is a standard measure of diagnostic concordance. And in general, you can see for classification of any of these entities, the concordance, the kappa score was relatively poor, indicating a relatively poor concordance. We were able to actually follow quite a few of our subjects to autopsy. And then we were able to obtain both an evaluation of the comparison between the PET measures and some measures of pathology, and also whether or not there was concordance between autopsy diagnosis and PET diagnosis. And in the left image, you can see this is looking at one cortical region, looking at the PET measure of amyloid burden versus autopsy rating of amyloid burden. And as you can see, this has quite a respectable Spearman rank order correlation. In terms of the overall kappa, comparing autopsy versus PET, the kappa score was 0.85, which is generally considered to be an excellent score. So this autopsy study validates this kind of multimodal PET method as a method for diagnostic classification. As a bonus, we had quite a bit of fluorodeoxyglucose data in these particular individuals as well. And in general, this multimodal PET tracer method seemed to be better than the FDG PET classification. Now, to return to an issue that we discussed previously, how can you use PET to try and interrogate pathophysiologically relevant aspects of disease? And mentioned previously that amyloid burden itself does not have a particularly good correlation with clinical features. On the other hand, tau pathology, in particular neurofibrillary tangle pathology, does correlate quite well with important clinical features of Alzheimer's disease. And this is just one example of a number of studies which have looked at correlations between neurofibrillary tangle burden and cognition in postmortem studies. And what you can see, particularly in the right graph, is there's a very tight correlation between a reasonable measure of neurofibrillary tangle burden and premortem measure of cognition, in this case, the mini-mental state exam. Now, it's generally considered that tau pathology is much closer linked to cognitive deficits than amyloid burden. And this is also consistent with a very popular notion that while amyloid may be a trigger for the development of Alzheimer's disease, tau pathology is really more likely to be the driving force in terms of neurodegeneration and the development of clinical features. Not surprisingly, these facts led to quite a bit of interest in trying to develop a PET tracer to quantify tau pathology in the brain. And this poses a number of significant challenges. So there is a single tau gene, but its biology is rather complicated. And in particular, the tau gene undergoes a significant amount of alternative splicing with the presence of different isoforms containing different amounts of exons. And particularly relevant seems to be the presence of how large number of repeat domains there are in the microtubule binding region of this protein. In addition, tau also undergoes a number of post-translational modifications, including different phosphorylation states, which are probably relevant to pathogenesis. And all of these different features, including the alternative splicing and the post-translational modifications, probably contribute to the presence of different types of tau that are present in the brain, some of which may be relevant for pathogenesis. And different tracers may potentially bind to different isoforms of tau, which makes interpretation in some cases of the tau binding somewhat complicated. Now, as an example of some of this complexity, it's very well known that different tau isoforms are expressed in different neuronal populations, and they tend to predominate in different diseases. So, for example, in Alzheimer's disease, there's both 3-repeat, 4-repeat tau, which is expressed in neurons in the form of paired helical filaments and neurofibrillary tangles. In contrast, in progressive supranuclear palsy, you may have 4-repeat tau, which is expressed in astrocytes. Or in frontotemporal dementia of the Pick's disease variant, you may have 3-repeat tau, which is expressed primarily in neurons. Now, a number of different structures were pursued as potential tau tracers. And not surprisingly, some of these fell by the wayside. So, for example, the compound that's in the bottom of the middle column was found to bind primarily to another target within the brain, and it's this off-target binding rendered it as a poor candidate as a tracer. Among these candidates, however, one particular compound, which you can see identified in the top right of the image, emerged as what appeared to be a relatively good compound for identifying tau burden within the central nervous system. This is one of the early demonstrations of using this particular compound, which became known as AV1451, to identify tau in the human brain. And in this particular study, this is a set of images derived from cognitively normal individuals and individuals with Alzheimer's disease, both of whom had prior imaging with an amyloid tracer to demonstrate that they had a significant amyloid burden. And what you can see is only the individuals with felt to have clinical Alzheimer's disease actually had a significant burden of tau in their brain. This kind of identification of tau with the PET tracer was also seen in some other conditions. So, for example, in this particular experiment, you can see some expression of tau in an individual who had traumatic brain injury, and also in individuals with mild cognitive impairment, felt to be a likely precursor to Alzheimer's disease. Now, this particular structure became widely used for experiments, and quite a few people underwent tau imaging as part of large cooperative studies, particularly the Alzheimer's disease neuroimaging initiative. And this is from data compiled in the Alzheimer's disease neuroimaging initiative project. In this particular study, what the authors have done is to classify the image data as along the same lines as used by Brock in his classification of tau pathology post-mortem. And you can see this is classified from stage 0, where there's a modest degree of tau in the temporal lobes, up through stage 4, where there's diffuse tau burden. And in this particular experiment, the classification in this sort of quasi-pathological categorization was paralleled by the clinical features that exhibited by the subjects and also by other biomarkers of Alzheimer's pathology. So again, quite consistent with the idea that this was identifying a relevant role of tau deposition in the central nervous system. Briefly to this earlier study, there's a nice illustration here of what might be sometimes a potential pitfall with use of PET tracers, and that is the issue of off-target binding. And alert viewers will notice that in the coronal sections based at the top of the second and fourth column, you can see some high levels of binding within the midbrain and then immediately lateral to the midbrain. And this is off-target binding in the choroid plexus and also within the substantia nigra. And one of the things that was discovered about this particular tau tracer was that it appeared to bind to neuromelanin-containing neurons. And this was confirmed in some studies using Parkinson's disease subjects. You can see here in these midbrain images of a group of normal controls and also some Parkinson's disease patients that there's reduced binding in the Parkinson's disease patients who, of course, have loss of substantia nigra neurons that express neuromelanin. As a further illustration of the use of this type of tau imaging tool, this is from a recent paper in which a number of different data sets were aggregated to look at different patterns of tau deposition in the brains of demented individuals. And what was assessed here was whether or not there are characteristic subcategories of Alzheimer's disease patients. And these particular authors, using some fairly sophisticated data analysis methods, determined that there's a number of different subpatterns of tau distribution in these individuals. And you can see this here in the second set of images where there's patterns where you see individuals, for example, who have sparing of the medial temporal lobes, or patterns where there's more selective involvement of some portions of the temporal lobe than others. In addition, they had some longitudinal data and were able to generate some evidence indicating that these kind of patterns were sort of stable over time, suggesting that these are sort of characteristic subtypes of Alzheimer's disease. Just to conclude, the tau imaging efforts have been successful in terms of generating a tracer that seems to function very well in the context of Alzheimer's disease. But there is, still some challenges and further research ongoing in trying to image characteristic proteinopathies. So for tau imaging, there are now efforts to develop second-generation agents which have improved specificity, and also hopefully to develop some agents which have isoform selectivity, so that it would be possible to reliably image not only Alzheimer's disease, but also diseases such as progressive supranuclear palsy that express different types of tau isoforms. There's also a great deal of interest in trying to extend these type of methods to image other kinds of proteinopathies, particularly alpha-synuclein and TDP-43. To date, this has not been successful and is likely to be very challenging, as the concentration of these particular proteins in the brains of affected individuals is significantly lower than the concentrations of tau or amyloid in Alzheimer's disease. And that poses a significant obstacle to trying to develop a successful imaging method for these particular proteinopathies.